Hello and welcome to the webinar. Downstream-of-Gene RNAs, or DoG RNAs, are the RNAs transcribed continuously beyond the host gene transcription termination sites. Due to their extended lengths, some DoGs can further produce DoG-derived chimera RNAs and circular RNAs. DoG RNAs have many unique properties and important biological functions. They are often associated with cancers and diseases. This webinar will cover what DoG RNAs are, DoG RNA Biogenesis, DoG-derived chimera RNAs and circular RNAs, DoG functions and DoG diseases, DoG RNA profiling, and research roadmap ahead. Normally, a protein coding gene transcription starts at the transcription start sites, or TSS, and ends after the transcription end sites, TES. The pre-mRNAs with still ongoing transcription is cleaved at the poly-A signal at the TES and gets polyadenylated. Cellular stresses can induce the production of downstream-of-gene transcripts, or DoG RNAs, continuously beyond the TES. The DoG RNA lengths are typically between 5 to 200 kilobase, with average size of 14 kilobases. The chromatin of the DoG regions is rich in histone marks related to transcription elongation, such as H3K36 trimethylation or H3K79 dimethylation. Some DoGs can extend the transcription further into the downstream gene referred to as read gene genes to produce chimera RNAs or other read-through RNAs such as circular RNAs. After their synthesis, many DoGs remain close to their sites of transcription, whereas others are released into the nuclear plasm. Both polyadenylated and polyadenylated DoGs have been detected at the similar levels. DoG products and downstream gene targets can be classified into several types. DoG pre-mRNA is the pre-mRNA made from the DoG host gene. DoG link RNA is a downstream non-coding RNA as the DoG target. Chimera RNA is produced when a DoG and a downstream read-through gene splice together into a chimera RNA. Chimera RNAs are mostly non-coding RNAs functionally, even if the downstream gene is a coding gene. DoG circular RNA is a downstream circular RNA as the DoG target. DoG RT circular RNA is produced when the downstream read-through gene transcript is back-spliced with the DoG RNA to form a circular RNA with both DoG and downstream gene sequences. A downstream gene can be in convergent direction with the DoG gene as a DoG target. The transcripts are antisense overlapping, causing transcriptional interference and downregulation. Let's look a bit closer at the chimera RNAs. A chimera RNA is produced when the upstream DoG transcription further reads through a downstream reading genes in the same direction. The intergenic distance is within 30 kilobase. The chimeric junctions tend to between the upstream gene's second last exon fused to the downstream gene's second exon, which is called the 2-to-2 rule. The The reading genes are usually short and GC rich. Chimera RNAs tend to use the same canonical splicing sites and exon-intron boundaries. Some chimera RNAs may retain introns. Unlike normal mRNAs that are exported into the cytoplasm, chimera RNAs are usually retained in the nucleus. Chimera RNAs function as long coding RNAs due to their nuclear localization and insignificant downstream protein translation. When the downstream gene is a circular RNA host gene, circular RNAs can be made from the DoG read-through RNA by back splicing. Circular RNAs are known to have regulatory functions such as microRNA sponges. If the back splicing of the read-through transcript is between a downstream gene exon and an upstream DoG host gene exon, a circular RNA made of these two genes is made. These type of circular RNAs are referred to as read-through circular RNAs or RT-circRNA. The primary reason for DoG formation is weakened or failed transcription termination. For normal transcription, there is not a specific transcription termination signal sequence for the RNA polymerase II to stop. Instead, the polymerase continues with the transcription, passing the actual transcription end sites TES. It is the cleavage and polyadenylation complex CPA that cleaves the pre-mRNA at the poly-A sites to release the gene transcript product. The exoribonuclease XRN2 then enters at the exposed 5' end and digests the downstream portion of the transcript still undergoing transcription. The fast XRN2 catches up with the polymerase like a torpedo to stop the transcription at the transcription termination site TTS. Normal transcription termination requires multiple coordinated events, including factors for RNA polymerase slowdown. For example, by spt5 dephosphorylation, histone modification, or histone variants, cleavage at poly-A sites, for example, by YSH1, cleavage and polyadenylation specific factor CPSF73, chase down and dislodging of RNA polymerase RNP2, for example by exoribonuclease XRN2, SEN1, and SENTEX1. Perturbation in any of these steps can impact on the transcription termination and lead to the transcription read-through to make the DoGs. Cellular stresses such as osmotic stress, heat shock, viral infections, or cancer senescence can all promote aberrant transcription termination. Under the stress conditions, many genes fail to terminate transcription properly and favor read-through transcription due to the failure or dysfunction of transcription termination factors. In addition, DoG-producing genes often have distinctive chromatin features that predispose them to read-through transcription. For example, enriched histone marks such as H3K36 trimethylation and H3K79 dimethylation not favor transcription elongation or weaker polyadenylation signals PAS. DoG formation is regulated by epigenetic and epitranscriptomic modifications. Histone H2AZ is a histone variant associated with DNA and repair. Normally, H2AZ histone can regulate polymerase II elongation rate, facilitate transcription termination, and inhibits DoG formation. However, in cell senescence, H2AZ occupancy is absent, allowing polymerase II to progress into the downstream genes to form family of senescence-triggered antisense residue RNAs referred to as start RNAs. The start DoG RNAs are antisense overlapping with the downstream convergent genes, inhibiting downstream gene expression. In addition to H2AZ, H3K37 trimethylation can also regulate DoG formation. In a variety of cancers, mutated H3K36 methyltransferase gene SETD2 causes dysregulation of H3K36 trimethylation, causing defects in transcription termination and extensive DoG formation, underlying this being a general cause of cellular transformation. SETD2 is a histone H3K36 methyltransferase. In clear cell renal carcinoma, ccRCC, SETD2 mutation leads to H3K36 trimethylation reduction, defects in transcription termination, extensive transcription residue, and DoG formation. Importantly, expression of Y-type SETD2 in ccRCC cell line deficient in SETD2 is sufficient to recover the defects in transcription termination in some DoG-producing genes. Epitranscriptomic modification of transcript can enhance transcription termination and restrict DoG formation. Here, number 1, M6A RNA modification at 3' UTR of gene 1 is recognized by the YTH DC reader domain of the cleavage and polyadenylation specific factor CPSF30L that promotes cleavage at the transcription termination sites. The cleavage at poly A sites assist RNA polyadenylation. 3. The facilitated transcription termination restrict the chimera RNA formation between gene 1 and gene 2. R loops are RNA DNA 3-stranded hybrid loop structure occurring in the genome. They are formed during the nascent RNA transcription. R loops can slow down RNA polymerase II, recruit syntaxing RNA helicase SETAX to release the RNA transcript, and allow exoribonuclease XRN2 to chase down RNA P2 to terminate the transcription, all of which suppress DoG formation. By overexpression of RNase-H that digests RNA in the R loop and disrupts R loops, DoG formation is increased. M6A epitranscriptomic modification is known to facilitate R loop formation, thereby slowing down RNA polymerase II. If M6A writer protein metal 3 and M6A modifications are deficient, the R loops are abolished, causing transcription termination defects, read-through transcription, and DoG formation. On the right, a methylpurine DNA glycosylase MPG can enzymatically remove M6A in the R loop and leave a basic site in the RNA. The basic site inhibits endogenous RNA-SH activity that degrades R loops to protect and increase the R loop levels. Thus, the stabilized R loop can pause RNA P2 and facilitate the transcription termination. In addition to M6A regulations of R loop, and DoGs by RNA polymerase slow down. M6A can also regulate R-loop and DoG formation by exon 2 exonuclease traced on RNA polymerase 2 for transcription termination. Here, RNA helicase DDX21 binds the R-loop and recruits M6A writer metal-3. The M6A reader YTHDC1 then recruits exoribonuclease exon 2 to trace the ongoing RNA polymerase 2 to terminate the transcription. In XDD21, metal-3 and M6A lockdown cells, exon 2 traced on and transcription terminations are defective, causing transcription read-through and DoG formation. During herpes simplex virus HES1 infection, the cellular stresses cause widespread disruption of transcription termination. Viral protein ICP27 binds cleavage and polyadenylation specific factor CPSF to form dysfunctional 3' processing complex, disrupting host gene transcript cleavage and polyadenylation and transcription termination. However, for viral proteins, the viral protein ICP27 binds specific viral sequence near but not on the viral polyase sites, acting as a 3' processing activator instead, so the viral genes are transcribed and terminated normally. DoG functions and DoG diseases are currently the focus of active research, with several proposed mechanisms highlighting their involvement in gene regulation. DoG transcription read-through and chimeric transcripts are prevalent in cancer. The transcriptional profiling of a cohort of 50 primary clear cell renal carcinoma (ccRCC) samples from the Cancer Genome Atlas (TCGA) collection shows that the invasion of the downstream labeling genes, aberrant expression of oncogenes, and generations of RNA chimeras as the functional outcomes of DoG read-through. For example, a DoG KDSR gene read-through is positively correlated with downstream BCL2 oncogene expression. B. In the tumor samples, the DoG read-through sequencing recounts are significantly higher than in the normal samples as shown in the yellow shaded DoG region. C. In the end, the high DoG read-through dramatically reduces cancer survival compared with the low DoG read-through cohort. If the downstream gene is in convergent antisense direction, the DoGs can act as an antisense RNAs to repress the downstream gene expression by transcriptional interference. Commonly in senescence cells, a family of senescence-triggered antisense read-through RNAs, or STOT RNA for short, are induced as DoGs. Importantly, STOT RNAs repress the expression of the downstream convergent RNAs. DoG read-through transcription can alter the chromatin structure of the downstream gene and thus the downstream gene expression. In influenza A virus infected cells, viral long structural protein NS1 induced the read-through transcription of highly active host genes, causes cohesion displacement from the host chromatin, eliminations of chromatin loops, and decompactions of chromatin in the read-through regions. The read-through into heterochromatin changes active A to inactive B compartment. About 5% of the attendant gene pairs in the human genome can be transcribed into precursor RNAs that eventually splice into chimeric RNAs. A remarkable feature is that some chimera RNAs are recurrently detected in different tumor samples. For example, chimera RNAs are pervasively transcribed in renal carcinoma. In particular, chimera RNA CTSC and RAB38 between cathepsin and RAB genes is observed in about 20% of the TCGA cancer samples, but not in any matched normal samples. In bladder cancer, chimera RNA CHFR and GOGA3 between checkpoints with forecat and ring finger domains and Golgi genes is observed at high frequency compared with adjacent normal samples. Interestingly, both their parent genes are not differentially expressed. In prostate cancer, chimera RNA SLC45A3 and ALK4 between SLU carrier and X transcription factor genes can be induced by androgen hormone by overcoming the read-through blocks imposed by CTCF insulator in the endogenic region. It is the chimera RNA, not the ALK4 encoded protein, augments prostate cancer cell proliferation. Here, chimera RNA acts and functions as link RNA, not as ALK4 mRNA. As ALK4 protein levels are not significantly different between the cancer and normal cells. DoGs are also biologically active in other kingdoms of life. In plant, CBF1 is a highly conserved transcription factor for plant co-response. Its expression is initially increased upon co-exposure. After 4-hour co-exposure, DoG gene SVK transcribes read-through antisense DoG RNA antisense to CBF1 gene downstream, storing the CBF1 expression by RNA P2 collision, thereby tightly controlling the expression and timing of CBF1 and co-response. Array-star human downstream-of-gene transcript or DoG RNA microarray is designed to profile and study human DoG RNAs. The array includes more than 13,000 probes to simultaneously detect and quantify DoG RNAs, DoG pre-mRNAs, DoG chimeras, and downstream transcript as their potential regulatory targets at a high accuracy and specificity. Array-star DoG RNA microarray probes are designed to target the unique sequences in the splice junctions in the DoG regions, DoG host genes, DoG chimeras, DoG read-throughs, and downstream link RNA and circular RNA targets. Compared with sequencing, DoG microarray is much simpler and more accurate, providing highly sensitive signals that detect and quantify DoGs and downstream target RNAs, including low abundant circular RNAs and multicircular RNAs. DoG profiling by sequencing will require costly deep coverage that remains inadequate. For DoG profiling, Array-star DoG microarray offers a unique, tailored solution. Here are the specs of Array-star human DoG array, simultaneously covering 4,460 DoGs, 4,460 DoG pre-mRNAs, and all their downstream link RNAs, downstream circular RNAs, chimera RNAs, RT circular RNAs, and downstream antisense transcripts. The DoG profiling data are analyzed for DoG differential expression with full change Fc and statistical significance p-values. The DoGs are annotated with detailed information about the DoG RNAs and DoG downstream targets. As a roadmap ahead for DoG study, total RNA samples in two comparison groups, for example, disease versus healthy, treated versus untreated, test versus control, are profiled by ArrayStar DoG microarray. The top differentially expressed DoGs can be confirmed by qPCR or in situ fluorescent hybridization FISH. To study the selected DoG functions, the DoGs can be locked down by RNA interference or CRISPR-I. Antisense oligo ASO can be designed to target RNA sites to induce RNA cleavage and transcription termination that prevent the DoG formation. Transcription inhibitors such as actinomycin D can be used to inhibit DoG transcription and to observe the phenotypes. To study DoG mechanism of action, DoG interacting protein factors can be identified by chromatin isolation, by RNA purification, and mass spectrometry, or TRIP-MS. DoG regulation by R loops can be explored by drip sequencing. M6A epitranscriptomic regulation of DoGs can be profiled by epitranscriptomic microarray or myriad sequencing. Nascent DoG transcription can be identified by mammalian native elongation transcript sequencing or NNAT-seq. Let's have a look at some cases of DoG research using these methods and approaches. Here, cathepsin and RAP genes DoG chimera, CTSC and RAP38 is studied by siRNA lockdown and qPCR analysis using the primers as indicated in Figure A. As shown in Figure B, CTSC and RAP38 chimera RNA can be locked down by chimera junction-specific siRNA, but not by its host gene-specific siRNA, showing the siRNA specificity and independent expression of DoG and host genes. In this study, the DoG of SRSF1 gene is locked down by CRISPR-I interference using single-guide sgRNA targeting the SRSF1 gene promoter labeled as IS9. CRISPR-I's line coordinately locked down SRSF1 gene pre-mRNA E1 and I1 and DoG1 and DoG2 regions. DoG functions can often be inferred by the functions of DoG RNA binding proteins RBP, in chromatin isolation by RNA purification CHIRP. DoG RNAs and the binding proteins are cross-linked, sonicated, and hybridized to biotinylated oligos. The complexes are pulled down by strobividin magnetic beads. After removing the RNA by RNase digestion, the enriched DoG binding proteins are identified by mass spectrometry, as in TRIP-MS. DNA sequencing can optionally provide genomic binding sites information. As an example, the most significantly enriched binding proteins with the highest fold enrichment and significance analysis of interacton SAMT SANT scores have been identified as small CD1 and BERT chromatin regulator proteins for the selected RNA in endoderm differentiation called digit. In this study, the genome browser tracks for MYC oncogene locus show the R-loop profile obtained by drip sequencing and nascent transcript profile obtained by NNAT sequencing. When M6A modification is reduced by M6A writer metal-3 lockdown, the R-loop signals are reduced, whereas the nascent read-through transcripts after the TES are increased, unveiling the DoG regulation by M6A promoted R-loop formation and facilitated transcription termination. I hope the information so far is helpful for your DoG RNA research. Thank you for your interest. Please visit us at arraystar.com.